This is our Olympus time-lapse system. It's a fully automated, inverted microscope surrounded by a heated chamber, and it allows us to image cells growing in a nutrient medium in plastic dishes and doing things over long periods of time. Every part of the system is automated and motorised and can be controlled through computer software. The software has a really easy to use graphical interface, and this allows us to program in the steps of a time-lapse experiment simply by dragging and dropping icons onto a screen, linking them together and wrapping other instructions around them. Here is a typical imaging experiment. We've already used the system to record a list of positions which we want to image during the experiment and save this list to a file called a stage list. Starting from the outside and working in, our time-lapse experiment consists of a cycle every 5 minutes, repeated 50 times, which we apply to all the positions stored in a stage list, and then at each of these positions, following the icons in the inner box from left to right, we do the following. First we wait for 50 milliseconds to let the system stabilise, then we do an autofocus to really optimise the focus before taking an image, then we take a transmitted light image with a 200 millisecond exposure, and then we take a series of 3 different fluorescence images to look at 3 different fluorescence dyes in our sample. Each of these images has a different exposure time, and since different colours focus at subtly different heights, we change the focus by a very small amount between each image. Once we've taken all the images at this position, we move to the next position, and the next one, through the stage list. Once all the positions in the stage list have been imaged, the system then waits until the whole experiment has been running for 5 minutes, and then it repeats the cycle again and again 50 times to do the complete experiment. Here we can see the imaging happening at a single position. The stage brings the position into view, pauses, and then takes a series of images to analyse for the autofocus. That's the transmitted light image, and now first, second and third fluorescence images using different colours of excitation, and then it moves to the next position. Many cells in our bodies are not inert little blobs that just sit there. Many are actively motile, moving around or continually exploring their environment. Let's take a look. Here we see the sheet of cells on either side of a scratched wound moving together to seal the scratch. This is effectively what is happening under a scab after you've cut yourself. Here we see a type of cell called a fibroblast, which, although these are not moving around very much, are very active, ruffling their membranes as they explore their environment.